Welcome to my channel. I am Lasya Priya. In this video, I want to explain a concept from vectors, which is Bac and Yac vectors. Now, let us know what a vector is. A vector, as related to molecular biology, is a DNA molecule, often plasmids or viruses, that is used as a vehicle to carry a particular DNA segment into a host cell as a part of cloning or recombinant DNA technique. The vector typically assigns in replicating and expressing the inserted DNA sequence inside the host cell. Vectors are used as a tool in molecular cloning procedures, so as to introduce the desired DNA insert into a host cell. Vectors are of two types: cloning vectors and expression vectors. We are learning about cloning vectors, which are divided into chromosomal and artificial chromosomal vectors. Artificial chromosomal vectors can be created by joining segments of DNA taken from cells of different species. Chromosomal vectors cannot be created using DNA taken from cells of different species. Chromosomal vectors are plasmids, cosmids, phasmids, and bacteriophage vectors. Now, we will learn about artificial chromosomal vectors. First, we will look at bacterial artificial chromosomal vectors. From a gene, we separate gene of interest and it is inserted in the vector. The vector with gene of interest is known as rDNA, also known as recombinant DNA. The bacterial cell or the host cell that take up the foreign DNA is called transformants, while the bacterial cell that doesn't take foreign DNA are non-transformants. The host cells are added to the media containing agar and chloramphenicol. When they are added, the host cells which are transformants, that is, host cells with gene of interest, can only survive in this media, whereas the host cells that are non-transformants cannot survive, therefore they die. Colonies with recombinant gene turn white color, and the colonies without recombinant gene turn red color. Just like how it happened in chloramphenicol, it also happens with SAGB. Here, the plus symbol represents the presence, as the minus symbol represents the absence. When the cell is transformed, the SAGB becomes inactive because the protein cannot be formed, due to which the sucrose won't be converted into 11 sucrose, which is toxic. So the cell can survive due to 11 sucrose doesn't form in the cell of transformants. In the same way, when gene of interest is absent in the host cell, the SAGP is active, which converts the sucrose into 11 sucrose. Due to the formation of 11 sucrose, the cell dies. The second chromosomal vector we are going to discuss is yeast artificial chromosomal vectors. Here we will see thymidine kinase. This is also called as false positive result method. In the same way as we done in Bac, the gene of interest is transformed into the host cell with the help of a vector. If the gene of interest is present in the host cell, then the thymidine kinase will be inactive, where the enzyme is not active. So, the cell will die. When the gene of interest is absent in the host cell, the thymine kinase is active, where the enzyme is active, so the cell will live. This method, we are getting a false result, so we can do it in a different way. Has been done in the back chlorophenol. We will add the host cells into the media that contains agar and amblycynical resistance. The cells with gene of interest can survive, and the host cells without gene of interest cannot survive, so they die. The next one is SUP4. When the gene of interest is present in the host cell, then the SUP4 will be absent in the cell. So the ADA2 doesn't get suppressed. So in this cell, ADA2 is active. This causes nucleotide synthesis and canonine is produced. Due to these activities, the cell appears in red. Whereas when the gene of interest is absent in SUP4 is active, which suppresses the ADE2, which doesn't synthesize nucleotide and canonine. Due to this, the cell turns into white color. I hope you understood the concept. If you have any doubts, please ask in the comment section. And thank you for watching.